Okay. I suggest that everybody takes his seat, and we will continue with the second part of the program. Just an information: at the end of this session, there will be lunch served outside, so you can concentrate on the lectures, and food will come later. It's really we have two lectures on this session. It's the Beutler awardees who will actually, the first two awardees who actually will present their science. And the first one is Professor Karl Skorecki. It's really my great honor and privilege to present. Professor Karl Skorecki is currently the director of the Rambam Family Institute of Research in the Medical Science and Director of Medical and Research Development at Rambam Healthcare Campus. I did personally had a chance to work with him as dean in the previous term, and now for the last more than 10 years we're actually working together. He was born, educated in Toronto, Canada. After receiving an MD in 1977 from the University of Toronto, he pursued postgraduate clinical research, training in internal medicine, nephrology, and molecular biology at Harvard Medical School in Boston, Brigham and Women's Hospital, and Mass General Hospital. In 1984, he returned to University of Toronto, where he went on to serve as chief of nephrology and professor in medicine, pediatrics, and clinical biochemistry. After a sabbatical at the Weizmann Institute in 1991, Karl, his wife Linda, and his family moved to Israel in 1995 and joined Rambam and the Technion Faculty of Medicine in Haifa. Karl's research main focus has been in population genetics, in human health and disease. He has received several prizes for his research and biomedical innovations, as well as university and international teaching awards. His research discoveries have been widely recognized nationally and internationally and attracted public and media interest. The research work of Karl and his lab colleagues and collaborators in the population genetic of kidney disease constitute the basis for receiving the first Ernst and Bonnie Beutler Research Program of Excellence in Genomic Medicine for his research project entitled Identifying the Target Strategies for Prevention and Therapy of Kidney Disease, Application of Population Genetic Discoveries to Advanced Experimental Research Platforms. I'm very pleased and honored to invite Karl, Professor Skorecki, to tell us more about this exciting research and its implication to our clinical life. Thank you very much, Karl.
Thank you, Rafi, for that warm and generous introduction. And thank you all. So it's really a privilege for me to be able to speak about work that's really a group effort, and you'll see the group in a moment. It relates to a particular genetic locus called ApoL1, which encodes apolipoprotein L1, and I will try to give an overview, some progress, and some plans for the future from gene discovery to disease mechanism. And I'd like to begin actually with the credits and acknowledgments, because there are very, very many of them. And the Beutler grant has enabled and opened up for myself and the lab very, very important collaborations. And collaborators are sitting here in the front row. Their pictures, and I introduced them at the beginning: Waldemar Popik, Pravin Singhal, Thomas Weedy. And there are also collaborations supported by other funding sources, mostly in genetic epidemiology and pathogen discovery. Alan Templeton, colleagues in Africa, Barry Friedman, Dawn Ganem in viral pathogen discovery, Derek Fine at Hopkins, and Saron Rosset in Tel Aviv. And David Friedman we welcome him here. He is one of the pioneers in the co-discovery of APOL1 and its association with kidney disease, and we thank him for coming to this symposium and being a workshop participant. The travel of these 4 guests from out of the country is supported by the Beutler Symposium donation, and I thank the Beutler and Bonnie and the family for enabling this. Most of all, I'd like to thank members of the lab, and here we are hard at work in the Hula Valley, bird watching, and there are many, and I hope I haven't left anyone out. Past members, many of them are sitting right here in the second row, some up higher. I probably shouldn't, I should read them all, but all the names, but I'm really, really very, very grateful for the opportunity to have such excellent people teach me along the way in this particular project and many others. There are also faculty colleagues with whom we work, such as at D. Salzburg here and others, I won't name them all, who give us a lot of advice, help, and collaborative input, and other collaborators who I might mention along the way. So I think that's probably the most important part, and for me, probably one of the greatest benefits of having received this grant. So let's, like many stories, if it's a good story, there should be like a trilogy, like Lord of the Rings or something. So there's Volume 1, Volume 2, and Volume 3. Volume 1 is actually the gene discovery, and after hearing Professor Beutler's lecture, there really is no more hide and seek. I think you just find things right away. But when we were working on this, and also David Friedman and his group, there was still a hide and seek component. So let's start with that part. And let me just back up, because it's all about kidney disease and nephrology. And actually March is International Kidney Month, and March 15th is International Kidney Day. So a quick review of the function of the kidney. Of course, the functioning filtering unit is the nephron, which has a filtering compartment, filters 180 liters a day of ultrafiltrate, a reabsorptive compartment, and kidney disease, including hypertension, which is mostly a kidney disease, either is a reduction or failure of glomerular filtration, this compartment, or some mismatch between what is filtered and what is reabsorbed. I forgot to mention here, I think there should be nephrology department, sorry, the nephrology department, which I'm part of. I have to thank members of the nephrology department for allowing me to do this research and working together with me. Okay, back to the kidney. And really, we mostly are concerned ourselves as clinical nephrologists to a great extent with glomerular filtration, because that's what leads many people to end-stage kidney disease and dialysis. So here we see a beautiful scanning in picture of the capillary loop that is the glomerulus, where this ultrafiltration takes place at the single nephron level. This is a naked glomerulus. The actual glomerulus is not naked. It's covered with this absolutely beautiful layer of what's called, made up of podocytes, a very specialized cell type, which I'm going to spend a lot of time talking about, which wraps itself around the glomerular capillaries and sends out these beautiful foot processes. And in transmission AM, cross-section, so this is the blood compartment with erythrocytes, plasma with water and solutes, and macromolecules, and the ultrafiltration process takes place across this filtration barrier, which separates plasma, water, solutes from proteins and other macromolecules in the ultrafiltration process. And here it is in higher magnification. These are the foot processes, which we saw earlier, of these beautiful podocyte cells and the slit diaphragm in between them. And a lot has been learned about the podocyte. There's actually a podocyte club, a podocyte conference. The 11th podocyte conference is going to take place in Israel in 2016. And many of the proteins that are important to the normal functioning of the podocyte have been unraveled, and to a great extent they've been unraveled by positional cloning in human disease. So more than 30 podocyte and glomerular basement membrane genes have been discovered by positional cloning of rare variants in rare monogenic diseases. And these have given very important functional insights as to how the glomerulus works in ultrafilters. So genetics shedding light on physiology, different than the tubule, where it went the other way. However, unfortunately, not many of these are actionable or druggable, And not that many are sites of common variants in GWAS, or population-based studies of chronic kidney disease. And that's too bad, because the majority of progressive non-diabetic chronic kidney disease of glomerular origin does seem to involve the podocyte and its injury and dysfunction. So here we see a beautifully intact cell body podocyte foot processes, and then in podocytopathies, which constitute a big part of kidney disease, these foot processes merge with each other, efface, and disrupt glomerular ultrafiltration. And as I mentioned, fewer than 5% can be explained by primary abnormalities in those gene products that were discovered by positional cloning. Now, is it a big problem? It is a very big problem, because if we think of chronic kidney disease, no one really knows the number, but it's at least a couple 100 million people. And kidney foundations have decided to stage kidney disease, and it's staged by the glomerular filtration rate, from rates which are nearly normal, but where there's some kind of other injury to the kidney, to progressively increasing severity of chronic kidney disease, culminating in what's called stage 5 chronic kidney disease, or end-stage kidney disease. The numbers are best known in the U.S. registries. Europe, Canada, and Israel are also well known, but these are U.S. numbers. So we see there are many, many people who have early-stage chronic kidney disease, millions, and fewer when we get to the pyramid of the more than 600,000 prevalent people on dialysis with end-stage kidney disease in the United States today. And is this because we're so good clinically as nephrologists at preventing the progression to end-stage kidney disease? Is this, a badge of honor? Unfortunately, it's a badge of shame, not a badge of honor, because the reason for this pyramid is because, unfortunately, most people with chronic kidney disease succumb, as has been shown in many wonderful epidemiologic studies, sad but important, because chronic kidney disease, the main problem with chronic kidney disease is cardiovascular morbidity and mortality rather than leading to end-stage kidney disease. So it becomes very important to try to understand this process. There's also a very important population ancestry disparity in chronic kidney disease incidence and prevalence rates, and it's been observed and now very well documented that African ancestry populations, and particularly first well documented in African Americans, have far more chronic kidney disease compared to other ancestries, Asian, European, for example, in populations in the United States and elsewhere. And this relates to percent African ancestry. So if we look at African Americans and add mixed population, I'll say more about that in a minute, then there is a relationship between percent African ancestry and incidence and prevalence rates. And with Hispanic Americans, which is a heterogeneous mixture of different degrees of African ancestry, but on average perhaps around 30% being intermediate in risk for these forms of chronic kidney disease. Now, this population disparity is actually evident for all the major etiologic categories of chronic kidney disease. And major etiologic categories are shown here, but it is particularly striking and interesting to us in the context of this project, in the context of non-diabetic chronic kidney disease in lean young adults with hypertension, attributed chronic kidney disease, non-diabetic glomerular disease, and others, and I'm going to say a fair bit about HIV nephropathy. In fact, the most striking discrepancy is in a form of kidney disease called HIV-associated nephropathy, where there is a very much greater fold risk for kidney disease of this type in HIV-affected African Americans compared to HIV-infected European or Asian Americans, where it's almost unheard of. So these population disparities have been studied for some time, and they're not readily attributable to socioeconomic, treatment access, or environmental factors. Moreover, there's an interesting familial clustering of chronic kidney disease and end-stage kidney disease of varying etiologies. So if there's an African-American dialysis patient, they will be more likely to have other members of the family with various etiologies of kidney disease compared to European ancestry patient with end-stage kidney disease. And this led to this thinking that there may well be an overarching population genetic risk variant or variance underlying this disparity, and which could shed light on the pathophysiology of some major forms of chronic kidney disease. We noticed something, and some of the names of the people who made these observations were on the slide of past colleagues and are authors of some of the papers you'll see here, that was brought to my attention, that Ethiopian Jews in Israel, and now we know this to be true in Ethiopia as well, have actually the low prevalence of chronic kidney disease and absence of HIV even when they have HIV infection. And we reported this just as an observation, and I'll come back to that. And so this notion from there must be an overarching renal failure susceptibility gene or genetic variance really pushed very hard by Barry Friedman, another Friedman who's involved in this whole area, with whom I also collaborate, to the discovery of DNA sequence variants in this APOL1 gene as being highly associated with the risk for many major forms of non-diabetic chronic kidney disease in African Americans, really was an effort of combined population genetics with evolutionary medicine. And I'll just briefly tell that story, because that story begins with a form of a disease, population-based gene disease discovery called mapping by admixture linkage disequilibrium. And the success of gene discovery using that approach depends on having an admixed population, so the African American population, I'll say a word more about that in a minute, having a disparity in the disease frequency between the parent populations that comprise that admixed population, and we just saw that for chronic kidney disease, and the postulate that there is indeed a causal risk variant or variance that are not rare, that have risen to high frequency in the at-risk parent population. That is something consistent with a common disease formulation. And that can happen unusually, and it happens under evolutionary selection, as we'll see. And so the principle behind GWAS and this form of GWAS is linkage disequilibrium. So I think we're familiar with it, but just quickly, if a marker is found in significant linkage disequilibrium with a disease risk causative variant, A, and here's the disease risk causative variant, we should detect association of marker A to the phenotype that variant predisposes to or causes. In GWAS, only those markers which are in sufficient physical proximity to the causative variant to mitigate recombination would be associated with a health or disease phenotype of interest. So meiotic recombination through the generations breaks down this linkage disequilibrium, and hence the genetic association to smaller and smaller and smaller regions. And admixture then restores the linkage disequilibrium genetic association to a larger block and really confers, I've simplified here, but to this kind of approach, the power of family-based linkage mapping, identifying large regions with greater facility and fewer markers. And that was the basis of this MALD approach applied to the African American population. So one uses markers which identify the ancestry of chromosomal regions based on that have very different allele frequencies in the parent populations, and uses that to discover those genomic regions with significant enrichment of the ancestry markers in the disease, in this case chronic kidney disease, compared to the control cohort, or even within the affected population to the rest of the genome. And in 2008, Cow and Kopp and others reported such a region on chromosome 22q1213. We also, with Liran Shlush and Doron Bahar and others, conducted a similar study, found the same region, reported a little bit later. And importantly, an important step was deconvoluting by removing diabetic nephropathy. So this was an association with non-diabetic forms of chronic kidney disease. After going after the wrong gene for a little while, finally, we and others, and I'll talk now mostly about this, honed in on this gene called apolipoprotein L1 in this region with about 30 different genes, first looking at an MYH9 for a while, for a couple of years, and variants were found in this APOL1 that had the very highest odds ratios and low p-values for association, lowest p-values for association with the phenotypes of interest. So this is the domain structure for apolipoprotein L1, And here are the kidney disease risk variants found. So the non-risk is, we call it wild type or G0, it's ancestral, and the derivatives are a missense haplotype, shown here, called G1, and a 6-nucleotide in-frame deletion called G2. And these were independently discovered by David Friedman, sitting here, with Martin Pollack and Giulio Genovese, and by members of my research group. And David has reported, and others, many studies have been done now looking at the allele, and this was in 2010, and genotype frequencies in African Americans. This is from David's paper in the Dallas Heart Study. And these are substantial allele frequencies. So we can see that fully in African Americans, 24% have one risk allele, 13% have two risk alleles. That is, 1 in 8 African Americans, and of course the number is higher in sub-Saharan continental Africa, have two risk alleles at this locus. Now, these, unlike many other GWAS studies, the odds ratios and the p-values for these associations with here's just 3 different etiologic categories of chronic kidney disease are very high. And this is taken from work by multiple groups shown here. So with HIVAN, it's odds ratios that are at least 29. Primary non-monogenic focal segmental glomerulosclerosis, 17. Usually GWAS studies give these odds ratios of 1.3, 1.2. Hypertension, probably the most common etiology, still has an odds ratio that's very high. That is a very large proportion of chronic kidney disease in African ancestry populations can be attributed to these variants at this locus, which makes it an important story in nephrology. It's also an exception to the rule, because we know that rare recent variants tend to be associated with a high risk for disease, and common variants, as I mentioned, tend to be associated with disease, but at not these high odds ratios. There are very few examples of common variants with large effect sizes, and here's one. These two variants, G1 and G2, in this genetic locus, APOL1. Now, even more interestingly, again, from a large multi-group effort, there may also be an association not only with kidney disease, but with an increased burden, this is a very recent study, of cardiovascular disease. So these are cardiovascular events that may be either independent of chronic kidney disease or maybe the earliest manifestation, something we probably will discuss throughout the day later and tomorrow. Now I want to go back, so the implications may be even broader, and I want to go back to the observation in Ethiopians, which for us was a very important opening and clue. I mentioned the low prevalence, and then here in a subsequent follow-up, having identified these loci, for a while, looking at other variants in the region, we found that there were substantial frequencies in Ethiopian populations who did not have HIVAN or this tendency to chronic kidney disease that sub-Saharan African populations had. But what distinguished Ethiopians was that they had a frequency of 0 of these G1 and G2 risk variants like Europeans and Asians. So the Ethiopian DNA markers reflect a different population genetic history, not surprising to population geneticists, and evolutionary selection pressure compared to Western Southern Sub-Saharan source populations, which are the source populations for African Americans. In other words, there's a phylogenetic split. Now, what is this evolutionary selection pressure, and what can we learn from it? And it relates to infection. That is trypanosomal disease, which can cause African sleeping sickness. So for years, APOL1 had been studied as the trypanolytic resistance factor by Pays and colleague and Raper and colleagues, and showed that circulating apolipoprotein L1 efficiently kills many, but not all, trypanosoma species. And it does so by hooking on to HDL3. The trypanosome imbibes it, and then it kills the trypanosome by punching a hole in the lysosomes. But there is one subspecies of trypanosome, Trypanosoma brucei-rhodesiense, which outwitted this conventional APOL1 by expressing something called SRA, which interferes with the ability of APOL1 to kill this particular trypanosome. And the G1 and G2 variants modify the protein structure at that C-terminal domain in such a way as to confer protection from this subspecies of Trypanosoma rhodesiense in sub-Saharan Africa. And Shai Tzur in the lab and others predicted that it would exert a disruptive effect on binding to the SRA domain. So it's spy versus spy. But better than predicting is the work that came out of Martin Pollack, Dave Friedman, and Giulio Genovesi, which showed that the sera derived from patients with these genotypes actually killed in the dish, in real life, these subspecies of Trypanosoma. And this was a very important finding. You can see the Trypanosoma dying there. So this explains, under selective sweep, the rise to high frequency on independent phylogeny twice in evolution, the G1 and G2 variants at APOL1, and a single allele is protective against the parasites. It also is what puts the entire genomic region under high linkage disequilibrium, something called hitchhiking, which is what enabled gene discovery via these tagging markers. And this is what has been permissive to these high odds ratios for common variants in a common disease. So that was the hide-and-seek gene discovery part. Now, there followed very quickly many genetic epidemiology studies, which have been important in understanding the scope of the problem, but also gave some clues and perhaps misclues regarding trying to go beyond, because this is all circumstantial so far. It's association. There's no causative proof yet. I haven't given any causative proof at all. And insights into mechanism. But one can try to infer something from some of these genetic epidemiology studies. There's so many by so many groups that I won't go over them all, but I will mention one or two that lead to volume 3. So there are multiple isoforms of ApoL1 without and with a signal peptide. Therefore, there's circulating and intracellular versions, and we can actually measure ApoL1 at the protein level and distinguish at the protein level whether someone is G1 or G2. Again, this is work from people sitting right here in the second row. And an experiment of medicine, not from our group at all. One is from a group with which Martin Pollack and David were associated, another from Barry Friedman's group. Experiment of medicine is kidney transplantation. So one can ask, does the risk travel with the kidney or with the recipient if there is a difference, and there is a difference, and it appears that the donor genotype associates with kidney transplant survival. That has practical implications already for kidney transplantation. It's being studied formally, but we've already had, even here in Israel, there are sub-Saharan African potential donors. They've come and we've actually rejected them as donors based on ApoL1 risk genotypes. But that needs to be studied formally. And it also may lead to mechanistic insights about risk traveling with the kidney, at least in part. So it's not all circulating ApoL1, maybe partly. So it's now much more than 10 studies across multiple etiologies. So another important observation show that the highest odds ratios occur under a 2-risk allele mode, so-called recessive mode, which would generally suggest, what, a loss of function type of mechanism. But there's a problem with that. So these are these high odds ratios, and these odds ratios occur under this 2-risk allele mode. And what is the problem? The problem is that ApoL1 seems to be dispensable in most mammalian species. So here's mice don't have any ApoL1. They have other members of the ApoL family. Humans do. Even among primates, chimpanzees have lost it. Gorillas and orangutans have ApoL1. Humans have ApoL1. And in a recent report from Duncan Johnson and others, was shown that there's a village, at least one, that's been studied in India where there's also a null state for ApoL1, and there's no evident kidney disease. Now, does that mean, so what does that tell us? Well, are there any associations with a single risk allele that would be consistent with some gain of injury? And the answer is yes, it probably is. It's mild. We reported one, namely the 2-risk allele mode leads to dialysis onset, accelerates progression to kidney disease 12 years earlier, but 1-risk allele, 5 years earlier, And also from the group in Boston, a report that it didn't reach significance because they did very stringent multiple hypothesis testing, but clearly an effect of a single allele also on time to progression. And there are several other examples of weaker associations under additive inheritance modes. So what is the formulation from a small single allele or additive effect together with the dispensability of APOL1 for kidney health? It suggests several possibilities, one of which is that the pathobiology, if it involves APOL1 and G1, G2, would involve a gain of injury model with a sharp APOL1 threshold effect due to multiple interactions of the gene product with endogenous or exogenous factors. And that's a formulation that one can certainly work on. And that brings me to volume 3, or the pathophysiology and the search for pathways and targets. And this is almost all related to collaborative work. And I have permission from the collaborators to show some of this work right now related to the pathobiology. And I believe some evidence that this is indeed causative and mechanistic insights. So again, what are the APOL1 disease associations? There is a group which are clearly podocyte diseases, focal segmental sclerosis, certain forms of lupus nephritis, namely the collapsing form of kidney disease, HIV nephropathy. These are clearly podocytopathies. There's a form of sickle cell kidney disease, which is clearly a podocyte disease. And there are disease entities in which the podocyte involvement might be there, but it is not as classic or well proven. That is hypertension-associated kidney disease, and this entity of cardiovascular disease with no overt reduction in glomerular filtration rate. Well, so if we use what's obvious, namely the podocytopathies, is APOL1 expressed in the podocyte. So as I hinted, APOL1, as you saw maybe on one of the earlier slides, APOL1 is sitting in a family of APOL1 through 6 genes in the human, which are contiguous and duplications of each other. And APOL2 is expressed at baseline in the glomerulus. APOL1 is hard to find at baseline in this filtering unit with the podocytes, the glomerulus, some controversy about that. But it is robustly and markedly induced in response to certain cytokines, and here interferon gamma at the RNA level, at the protein level. Sharon did these studies. And this is indeed consistent with the need for an exogenous stimulus or a second hit in the setting of APOL1 genetic susceptibility. So what else can we infer about the expression of APOL1 in the kidney? Well, glomerular APOL1 expression is also observed to be elevated in kidney biopsies with glomerulopathies in African Americans, but the correlations are not consistent and the numbers are very small. And what is being planned is an analysis of several hundred samples as part of an NIH-sponsored H3 Africa consortium, which our lab is part of, with the PI being in Ghana, Professor Adu. And here, even in non-African populations, this is from Thomas, there is modulation of expression of APOL1 and APOL2 in kidney disease in non-African subjects, suggesting that there may be lessons learned from the locus related to other forms of kidney disease, not only in sub-Saharan Africa. So again, where are we to take us in the pathobiology? The most prominent APOL1 disease associations are bona fide podocytopathies. And indeed, podocyte injury is thought to contribute to the progression of many forms of chronic kidney disease. The expression of APOL1 and other APOL family members is elevated in kidney biopsies of patients with glomerulonephritis, but that needs a lot more study. APOL1 expression in podocytes and also other cells, as I'll show you in a minute, is induced by inflammatory and immune mediators. So this induction is a whole subject area, namely the regulation of APOL gene expression, and that's what David Friedman is focusing on, and probably many other things, but he's going to talk to us about that. And model systems for podocyte injury, which I'm going to talk to you about, and then very basic cell biology and biochemistry that can be learned in many surrogate systems. And I think a key here to understanding is this relevant to the association, and is what we're studying important, is to see if there is a difference between the non-risk G0 and the risk variance. If there is a difference in whatever we're studying, it's probably relevant and important. So this is a collaboration with Pravin Singhal, in which APOL1 lentiviral transfection indeed causes a concentration-dependent podocyte swelling and injury with a nice threshold effect. So you can get down to a lower dose where the wild type, this is a measure of swelling of podocytes in culture. So this is human podocytes in culture. This is the system being used. And we see a very marked difference between G1 and G2 compared to G0 at higher concentrations of APOL1. Even the wild type has some noxious effects on human podocytes in culture. And this is just other measures of podocyte injury. Apoptosis is hardly affected, but swelling and then fragmentation is markedly affected, and importantly, with a marked difference of the G1 and G2 compared to the wild type, lysosomal leakage with cathepsin L, and cytoskeletal disruption. Additional features of APOL1 injury in the podocyte transfection model system, again with Pravin, augmented genotype-dependent susceptibility to injury with known second hit toxins. So reactive oxygen species, TNF-alpha, a classic animal model for acquired podocyte injury is puromycin administration, and HIV all cause a augmentation which is much greater in the risk compared to the non-risk variants. Again, work with Pravin Singhal. And injury from conditioned medium as well. So just going back, remember we have these two forms, a secreted and intracellular form, and we can see it in cells. This is in the interferon stimulation. We can see the form without and with the signal peptide, and therefore these forms occur naturally. And then the question is, which of them is actually causing injury? And that's why these conditioned media experiments are so important and require further study. APOL1 is the only member of the family that has a signal peptide and that gets out. APOL2, for example, doesn't, and APOL1 is also distinguished from APOL2 by having a BH3-only domain. So this leads to the current studies, a collaboration with Thomas and the group in Munster, namely doxycycline-inducible constructs, which are being generated now using gateway technology with Thomas and his group, in order to get specific expression of APOL1, wild type, G1 or G2, into podocytes. And this works. So here we see that minus plus doxycycline. We can see the different forms, isoforms, being induced. So we have actually two constructs made by Thomas, one with a GFP in place of the signal peptide, and one with a native. And very nice doxycycline-inducible expression of both isoforms. And so far, no effect seen with the inducible expression of the wild type in podocytes. We're now making the risk variants and plus-minus second hits to see if we do, in fact, see a phenotype, a cellular phenotype with this model. Now, that's cell culture. I learned from Bruce, and we learned today, but I learned it when I went to visit him, that it's important, especially when you have this complexity of circulating cells. You can't just look at one cell type and hope for the best. It's important to use organism models as well. And the first stab at this was actually transient transduction of the risk variants versus the non-risk variants versus hydrodynamic gene delivery transiently in mice, and indeed there was a gain of injury phenotype. So when mice were exposed to, by this method, to APOL1 wild type versus G1 versus, again, a non-risk variant, there was a clear effect of the risk variant compared to the non-risk variant in inducing proteinuria in the mice. And when the kidneys were harvested, we see indeed that the control compared to the G1 wild type or G1, wild type does not induce an effect on the podocytes, but the G1 causes this classic effacement of podocytes. But that is not a very easy to use model, and the better model would be to knock in or use a transgenic technology for murine-inducible expression of APOL1 risk variants, and this is ongoing work, again, in collaboration with the colleagues who have come here from afar today, either using the Cree-Lock system with an APOL1 promoter or using a TET inducible system. I won't go into the details, but obviously the advantages, because we don't have any results yet, we're just building these, the advantages of organismal models are that one can examine the differential effects in the context of either intercellular or circulating APOL1 non-risk and risk under conditions mimicking human expression and second hits in a whole organism. So mice have no endogenous. We put in the human non-risk and risk and see what happens. And the other organism model we're working on, and Adi Salzberg is helping us with that. We have a number of Drosophila geneticists in the faculty. It turns out that the fly has a very interesting excretory system in which the glomerular component is not connected to the tubule component, but rather than podocytes, there are nephrocytes, and the ultrafiltration process takes the nitrogenous waste products, puts them into a structure, and then the fly dies, so it's not actually excreted, and the tubule component just deals with salt and water and electrolyte balance. And this has been studied in terms of podocyte biology using the fly nephrocyte. And indeed, with Adi, one can see here there are actually two kinds of fly nephrocytes, so-called pericardial and Garland nephrocytes, and one can see them expressed, Tali Shemer did this, and in the fly using staining methods, etc. And now what we've just done is then use Drosophila genetics, which is a powerful tool, using driver flies and then transgenes which are driven with either the non-risk or risk variants of APOL1, G1, G2, or with a second hit, and either with ubiquitous expression, mesodermal drivers, or nephrocyte-specific drivers. And right this morning, I just got from Pazit News that we just set up these experiments, and indeed there is a fly phenotype with the risk variants. And it's when the risk variants are expressed under a ubiquitous expression driver. It's a winged phenotype. This is fresh as of this morning verbal. And with G2, an even more severe variant where the pupa, or the flies are stuck as pupa. And now, obviously, mesodermal driver does nothing. The G0 doesn't cause a phenotype. And obviously, one needs to do this in nephrocyte and other specific drivers. So I think we are on the verge of having important organismal model systems. I think there's a clear difference in cell culture and a number of models now between G1, G2, which makes us confident that this is an important pathway. Now, what about the actual detailed cell biology? I showed you the BH3 domain, and there are two major well-studied there are more, but two that are very well-studied cell biologic processes crucial for the contribution of podocyte integrity to glomerular permaselectivity. One is endosomal trafficking, which is rapid, and there's fluid phase and receptor-mediated trafficking, and this was described back in 1960 by Marilyn Farquhar and Palladio. And autophagy, because podocytes are post-mitotic, highly differentiated cells, and knocking out autophagy components such as ATG5 really hurts podocytes because they don't self-renew and they depend on autophagy for lifelong health. So this is a long-term, age-related type of phenotype in the mouse. A core complex in both of these processes is the VPS34-15 Becklin-1 core complex. And the question is, does APOL1 have anything to do with this at the cell biology and biochemistry level? And I go back to this domain structure with the BH3 domain in APOL1, but a corrupted BH3 domain in APOL2. And indeed, in collaboration with Waldemar Popek, again, I have his permission to show this, APOL1 induces a striking inhibition of Becklin-1 ubiquitination, and it's a much stronger effect with G1, wild-type G1 and G2 in this system. So this shows ubiquitinated Becklin-1, and this is total Becklin. So this is with HA ubiquitin tag. Again, I won't go into all the details. We have a workshop this afternoon, and it's part of my more detailed scientific progress report. And we can see the very, very striking inhibition of ubiquitination of Becklin-1 with APOL1, and this is in HEK cells. And also accompanying that is a disappearance of VPS34. So vector transfection of HEK cells, G0, G1, G2, much stronger disappearance of this vacuole sorting protein 34, part of that complex that I mentioned. So now the challenge is to look at if there's an inhibition of Becklin ubiquitination, and that's associated with a known complex related to these two processes of endocytosis and autophagy, is to try to identify the E3 ligases involved and the nature of the ubiquitination, which lysines, what kind of ubiquitination, is it degradative, non-degradative, and so forth, and that's ongoing work which is progressing. And leads to an actual model, just ignore all this part, in which we can test the idea that ApoL1, via inhibition of Becklin-1, a polyubiquitination, so without ApoL1, with ApoL1, G1 and G2 more than G0, ultimately results through this complex in a reduction of VPS34 and its disappearance. Is that important? It seems to be very important in kidney health and podocyte homeostasis, a paper published in 2013 from a group including Tobias Huber, showing that indeed targeting VPS34 shows the importance of endocytosis and podocyte homeostasis, and these mice suffer a kind of kidney injury that is reminiscent, it's a podocytopathy, either through endocytosis defects or through autophagy defects, one rapid and the other slow. So what further studies are planned and underway? Obviously, we want these biochemistry, cell biology studies, which have been done in an easy system like HEK293 cells, need to be looked at now in podocytes. I see the Greek is not Greek here. That is, and we know that interferon gamma, which induces ApoL1, also mimics VPS34 degradation, but that could be an off-target effect of interferon gamma, and one needs, and these human podocytes are non-risk ApoL1, so that needs to be studied with ApoL1 variant induction with doxycycline, and by giving interferon gamma and eliminating the effect of ApoL1. Whole organism platforms, studies need to be done, and we have the tools now to do that, or they're underway. And then I've been very podocyte-centric here, and we are, but there are clearly, and this is work, very exciting work published just recently from Waldemar Popik's lab, showing that ApoL1 has a lot of effects, important effects in innate immunity, and it has a major effect in restricting HIV infection, work that he's going to talk about this afternoon, so I won't go into it. Very exciting work that obviously could easily relate to the kidney disease, especially in HIV. So the path from genetic disease to kidney, from genetic risk to clinical disease. There are 38 million African Americans. 5 million approximately have the 2-risk genotype. Fewer than 1 million will actually get ESKD, end-stage kidney disease, due to this ApoL1 risk. Of the 4 million who do not, some are censored, as we saw, unfortunately, due to mortality prior to end-stage kidney disease, and in some, there is no phenotype, no disease phenotype. An exception is HIVAN. So HIVAN in whites or African Americans with 0 or 1 risk alleles doesn't occur, even if they're undertreated or have undertreated HIV infection. True in Africa as well. That shouldn't happen, of course. ApoL1, 2 risk alleles, much greater than 50, perhaps 100% if they're undertreated, but with antiretroviral therapy in time, this abrogates HIV-associated nephropathy. So HIV is clearly an important trigger for kidney disease, but in whom? In those individuals who have this ApoL1 genetic risk. So the challenge then, that's a small part of the whole spectrum. The challenge then is to identify additional known or novel non-HIV viral pathogens or other triggers which cause kidney disease in those who are genotypically at risk but don't have HIV. So 2 overall strategies then in ApoL1 genetic susceptibility and perhaps other loci in other populations. One is to protect the podocytes, which are rendered vulnerable with this risk genotype, by identifying those specific pathobiologic interactions and targets, which I hinted at and we're studying, or identifying, preventing, and treating these second-hit triggers, like HIV, and that's this project we're doing with Dawn Ganem and Barry Friedman, funded by the National Science Foundation. So back to genetics. So one, as just in basic genetics, we have, I think, a slightly different view now on this vexing problem, we heard a much more elegant view of the question of heritability, missing heritability, which has been a vexing problem in GWAS studies, keeping in mind that we each harbor not only mutations, but chronic productive viral infections, sometimes often organ system specific. So there's a genome-virome interaction, and the effect of viral infection is host genotype dependent, as we've heard, and the calculation of effect sizes, penetrance, heritability, should be done in the context of a virome in the case of genetic susceptibility, and here's the example, APOL1 and HIVAN. And indeed, just last year, in an article in Science and a major push in the US and the NIH, is to try to identify mechanisms for chronic non-communicable diseases that are susceptible to vaccine. So the idea would be to genotype for susceptibility, identify at-risk individuals, and prevent these second hits if and when they can be identified. So again, this work has been enabled to a very great extent by the grant support of the Ernest and Bonnie Beutler program, and I thank them for it, and I thank everyone who's involved, Bruce, Jacob Rowe for bringing it, Mrs. Bonnie Beutler, the members of the Scientific Advisory Committee, Rafi Biar, Professor Apoloig, Jacob Rowe, Chaim Cedar, David Wallach, and Bruce Beutler, and all of you for listening, and especially my colleagues here and members of the lab. Thank you very much.
So we know that that there is HIVAN that exists in Asia, and there's obviously no APOL1 in Asia. So you think that that there are other genes also?
Yeah. So Dr. Wasser is asking about HIV-associated kidney disease. I'm not sure that's been shown to be HIVAN. Well, that's there are forms
Some of it's
immune
HIVIC.
complex kidney disease, but it's not collapsing with tubular cystic dilation. There are certainly other forms of HIV-associated kidney disease which are not HIVAN. So to the best of my knowledge, HIVAN is restricted to G1 and G2 individuals in sub-Saharan Africa, not present in Asia. It certainly is possible that there are other loci in other APOL1s or elsewhere which render the kidney susceptible to HIV-triggered glomerular, but not studied, and no admixed population has been looked at in that regard. And of course, hard to study in mice since you put in it.
Any further questions? If not, Dr. Surya Asadi.
Dr. Asad, Chief of Nephrology, Rambam.
Yeah. You can just state your, make it loud. Wait for the microphone so that people can hear
it. Again, it's not boring to give this lecture again and again and see all the advances. And after all, you are our teacher. And I want to ask you, at this point, do you believe there's hypertensive nephropathy, or it's just an association between hypertension and chronic kidney disease?
Okay, I'll go back to the mic. So, everyone heard Dr. Asadi's question. And I think there are probably two forms of the association of hypertension with kidney disease. So, a form in which there's renovascular problems, either microrenovascular, probably more common in elderly, European ancestry populations, Middle Eastern populations perhaps, and a form in which hypertension, and that can be helped, where the progression of chronic kidney disease can be slowed and helped, perhaps, not fully proven, by addressing the hypertension. It's pretty clear from many studies that the form of hypertension that many African Americans get, and which is, and this is an editorial that Dr. Wasser and I wrote, hypertension misattributed chronic kidney disease, which is actually chronic kidney disease first and hypertension as an early manifestation, at least in that subgroup of patients. I think a number of studies have pretty well shown that. So, and in those subjects, treating the hypertension doesn't slow the progression of chronic kidney disease, unfortunately.
Thank you very much, Karl.